All right, let's jump right in. And today we're gonna take a deep dive into glycogen metabolism.
Sounds good.
And we're talking about a really important process here.
Yeah, definitely.
For energy storage and release and all that good stuff.
Yeah, it's like the energy bank of our bodies, right? We're
Yeah.
constantly making deposits and withdrawals.
It's like that rainy day fund, you got to have it.
Exactly.
So we're specifically focusing on glycogen metabolism in muscle and liver tissues.
Yeah, those are the key players when it comes to glycogen.
They're like the VIPs of the glycogen world.
They are, each with their own unique roles to play.
Absolutely. So let's start by kind of setting the stage here.
Okay, sounds good.
What exactly is glycogen and why is it so important?
Well, glycogen is this big branch molecule made up of a bunch of glucose units all linked together.
It's like a giant tree.
It is, with all those branches.
And all those glucose units are like little packets of energy just waiting to be released.
And it's a really efficient way to store glucose.
Exactly, because, glucose is our primary energy source, right?
Right, especially for tissues like the brain and muscles.
So it's like having a readily available energy reserve right there in our cells.
Exactly. When you need a quick burst of energy, your body can tap into those glycogen stores.
Like when you're sprinting to catch the bus or, lifting something heavy.
Exactly. In those moments, you need energy fast, and glycogen is there to provide it.
Okay, so we've got this glycogen stored up.
Yeah.
Now, how do we actually break it down when we need that energy?
Well, it all starts with an enzyme called glycogen phosphorylase.
Okay, glycogen phosphorylase.
It's the key player in glycogen breakdown.
So it's like the key that unlocks those energy stores.
Exactly what it does is it cleaves off glucose units from the ends of those glycogen branches.
Oh, so it's like a lumberjack chopping wood?
Kind of, but instead of an axe, it uses a phosphate group.
Ah, so it's a phosphate-powered lumberjack?
You could say that.
And the result is glucose 1-phosphate, right?
Right, that's the form of glucose that's released.
And that's already prepped and ready for further metabolic action.
Exactly. It can be easily converted into glucose 6-phosphate, which is a central molecule in energy metabolism.
And from there, it can go to glycolysis and generate ATP.
Exactly. That's the whole point of breaking down glycogen, to get that ATP energy.
All right, so we've got glycogen phosphorylase chopping away at those branches.
But there's a catch.
There's always a catch.
Glycogen phosphorylase can only work on those straight chains.
Oh, so what happens when it hits a branch point?
That's where the debranching enzyme comes in.
Okay, the debranching enzyme, another superhero of glycogen metabolism.
It has two jobs, actually.
Two jobs, busy enzyme.
First, it moves a small chain of glucose units from the side branch to a nearby longer chain.
Okay, so it's like rearranging the branches a bit.
Exactly. And then it snips off the very last glucose unit at that branch point.
So it's like a two-step process.
It is ensuring that no glucose unit is left behind. It is. And all those released glucose units can then be converted into glucose 6-phosphate and enter the energy pathways.
Okay, this is all making sense now. So we've seen how glycogen is broken down.
Yeah.
But what about building it back up? How do we replenish those glycogen stores?
That's a great question. And it's a process called glycogen synthesis.
Glycogen synthesis, building those energy reserves back up.
Exactly. And it's not just a simple reversal of glycogen breakdown.
Oh, of course not. It's going to be more complicated than that.
It is. It involves a slightly different form of glucose called UDP glucose.
UDP glucose, what is that?
You can think of it as glucose with an energy backpack attached.
An energy backpack. I like that analogy.
It's like glucose is all geared up and ready for action.
So this UDP glucose is the building block for glycogen synthesis.
It is, and the key enzyme here is glycogen synthase.
Glycogen synthase, another important enzyme. I'm seeing a pattern here.
There are a lot of players in this glycogen game.
That's for sure. So glycogen synthase is the one that adds those UDP glucose units to the growing glycogen chain.
Exactly. It forms those specific bonds that link the glucose units together.
It's like a construction worker building a brick wall.
I like that, adding those bricks one by one to make a strong structure.
But we can't forget about those branches, right?
Of course not. We need those branches for efficient glycogen storage.
So there must be another enzyme involved.
There is. It's called the branching enzyme.
Okay. The branching enzyme, what does it do?
It creates those branch points in the glycogen molecule.
So it's like a tree surgeon making sure the branches grow in the right direction.
Exactly. It transfers short chains of glucose units to create those branch points.
Oh, so we've got glycogen synthase building those straight chains and the branching enzyme creating the branches.
Working together to make a nice, compact, and efficient glycogen molecule.
A real team effort.
It is, and it's amazing how this whole process is so finely tuned with specific enzymes for each step.
It's like a well-oiled machine.
It is, but it's not just a constant cycle of building and breaking down glycogen.
There must be some way to control when each process happens, right?
Absolutely. Both glycogen breakdown and synthesis are tightly regulated.
To make sure our bodies aren't wasting energy doing both at the same time.
Exactly. It's all about maintaining that delicate energy balance.
So we've covered the basics of how glycogen is built and broken down.
But how does our body actually control these processes?
What signals tell those enzymes when to get to work?
That's where we get into the really fascinating world of regulation.
Oh boy. Things are about to get complicated.
Maybe a little, but it's what makes glycogen metabolism so interesting.
I'm ready. Let's dive in.
All right, so one of the key regulators of both processes is an enzyme called phosphorylase kinase.
Okay, phosphorylase kinase.
It's like the foreman on a construction site, giving the green light for either demolition or building.
So it controls both breakdown and synthesis.
It plays a role in both by regulating glycogen phosphorylase.
That's our trusty lumberjack enzyme.
It is, and what's really interesting is that phosphorylase kinase itself is regulated by a cascade of events triggered by hormones.
Hormones. Those chemical messengers that control so much in our bodies.
Exactly, and the two major players here are epinephrine, also known as adrenaline.
Our fight or flight hormone.
Well, right, and glucagon, which is released when blood glucose levels drop.
So both of these hormones ultimately lead to the activation of phosphorylase kinase.
Exactly. And that sets off the cascade for glycogen breakdown.
So whether we're facing a sudden threat or just haven't eaten in a while.
Our bodies have a way to quickly mobilize those glycogen reserves.
Get that glucose flowing.
Exactly. And this is just the tip of the iceberg when it comes to regulation.
There are layers upon layers of control.
Involving other enzymes and molecules that fine-tune these processes even further.
Oh boy. This is already getting incredibly complex.
It is, but I think we've laid a good foundation here.
Yeah, I'm starting to see how everything is connected.
It's like a finely tuned orchestra with each instrument playing its part at just the right time.
And those hormones are the conductors.
Exactly, guiding the whole symphony of glycogen metabolism.
Well, before we get too overwhelmed, let's pause here for now and take a breath.
Okay, that sounds good.
We'll pick up our exploration of this intricate world in the next part of our deep dive.
Looking forward to it.
Get ready for even more fascinating insights into the regulation of glycogen metabolism.
It's going to be a wild ride.
All right. See you then.
Okay, bye.
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 Welcome back.
All right, so last time we talked about how glycogen phosphorylase gets activated by that whole cascade triggered by epinephrine and glucagon.
Right. Those hormones are all about mobilizing those energy stores when you need them.
Exactly. But, what about building glycogen back up?
Glycogen synthesis.
Yeah.
It's the flip side of the coin, right?
Exactly. And I'm guessing it's regulated in a pretty similar way.
It is, but with a slightly different strategy.
Okay, I'm intrigued. Tell me more.
So remember glycogen synthase, that enzyme in charge of building glycogen?
Yeah, the one that adds those UDP glucose units to the growing chain.
Exactly. Well, it turns out that glycogen synthase is actually inhibited when it gets tagged with a phosphate group. Oh, so it's like the opposite of glycogen phosphorylase, which gets turned on by phosphorylation.
Exactly. It's all about those phosphate groups acting like little switches.
Flipping those enzymes on or off.
Right, and one of the key players in this inhibition is an enzyme called glycogen synthase kinase 3.
Glycogen synthase kinase 3. Try saying that 3 times fast.
It's a mouthful, I know.
But I'm sensing a pattern here. These kinases seem to be really important in regulating these processes.
You're picking up on something crucial here. Kinases are like these molecular switches that can turn enzymes on or off by adding those phosphate groups.
And in this case, glycogen synthase kinase 3 is the one putting the brakes on glycogen synthase.
Right. It's all about making sure your body isn't wasting energy building up glycogen when it needs to be breaking it down.
Right. It's all about balance.
Exactly. And to make things even more complex, there are actually several other kinases that can phosphorylate glycogen synthase.
So it's not just one kinase calling the shots.
It's more like a team of regulators working together to fine-tune glycogen synthesis.
Wow, this is getting complicated.
It is, but that's what makes it so fascinating.
Okay, so we have these kinases, like glycogen synthase kinase 3, that put the brakes on glycogen synthesis.
Right.
But what tells them when to do that? What triggers their activity?
Remember those hormones we talked about earlier, epinephrine and glucagon?
The ones that promote glycogen breakdown.
Exactly. Well, they don't just promote glycogen breakdown. They also indirectly lead to the activation of glycogen synthase kinase 3.
Oh, so they're multitasking.
They're promoting breakdown on one hand and inhibiting synthesis on the other.
It's all about coordinating those opposing processes.
Yes, making sure the body's energy needs are met.
Okay, so we've got these hormones pushing for glycogen breakdown and inhibiting its synthesis.
Right.
Is there anything that works in the opposite direction, promoting glycogen synthesis and putting the brakes on breakdown?
You probably already guessed it, it's insulin.
Insulin, our blood sugar superhero.
It plays a huge role in regulating glucose metabolism, and that includes glycogen synthesis.
Okay, so how does insulin fit into this whole picture?
Well, it has several key actions. First, it inhibits glycogen synthase kinase 3.
Taking the brakes off glycogen synthase.
Exactly, allowing it to do its job and build up those glycogen stores.
Okay, that makes sense.
Second, insulin promotes the uptake of glucose into cells, particularly muscle cells.
Providing the raw materials for glycogen synthesis.
Right, you need glucose to make glycogen.
Of course.
And there's a third important action. Insulin activates another enzyme called phosphoprotein phosphatase 1.
Another enzyme, I'm starting to realize just how many players are involved in this whole glycogen story.
It's a complex process with a lot of moving parts.
So what does this phosphoprotein phosphatase 1 do?
It has a crucial role. It removes those phosphate groups from glycogen synthase, essentially reversing the action of glycogen synthase kinase 3.
So it's like an undo button for glycogen synthase.
Exactly, taking off the brakes and letting it do its thing.
So insulin is working on multiple fronts to ensure that glycogen gets synthesized and stored when glucose is plentiful.
Exactly. It's all about coordinating those energy storage and release pathways.
This all makes perfect sense for muscle tissue, which needs glycogen for its own energy.
Right.
But what about the liver?
Ah, the liver.
Remember, it's not just using glycogen for itself, it's responsible for maintaining blood glucose levels for the whole body.
That's right. The liver is like the body's glucose pantry, stocking up when times are good and sharing with other organs when they're running low.
So how does it balance glycogen breakdown and synthesis to keep everyone else happy?
Similar to muscle tissue, the liver also responds to those hormonal signals from glucagon promoting breakdown and insulin promoting synthesis.
Okay, that makes sense.
But there are some liver-specific nuances to how this regulation works.
I was just thinking that there must be something unique going on in the liver to make sure it can share its glucose when needed.
You're right. One key difference is in how the liver controls phosphoprotein phosphatase 1.
That enzyme that removes phosphate groups and activates glycogen synthase?
Exactly. In the liver phosphoprotein, phosphatase 1 is regulated by another protein called glycogen-targeting protein.
Glycogen-targeting protein.
We'll call it GL for short.
GL, all right.
So GL is like a guide directing phosphoprotein phosphatase 1 to its target glycogen synthase.
Okay, so it's bringing those two together.
Exactly. And here's where it gets really interesting. Glucose itself can actually bind to GL.
Wait, so glucose is directly involved in regulating its own storage?
It makes sense. If there's a lot of glucose around in the liver, it means there's plenty to store as glycogen.
So high glucose levels promote glycogen synthesis.
Right. And when glucose binds to GL, it causes a conformational change.
Shapeshift.
Exactly. And that shapeshift allows phosphoprotein phosphatase 1 to do its job and activate glycogen synthase.
So it's like glucose is giving the green light for its own storage.
It is, but what about the opposite scenario when glucose is scarce?
What happens then?
When glucose levels are low, GL doesn't bind to glucose and phosphoprotein phosphatase 1 stays stuck to the glycogen particle.
Keeping glycogen synthesis in check.
Right. Meanwhile, glucagon steps in, signaling for glycogen breakdown to make sure there's enough glucose to go around.
So it's like a constant tug of war between these opposing forces, all trying to maintain that delicate balance of blood glucose.
Exactly. And that balance is absolutely crucial for our overall health and well-being.
So we've talked about these hormonal signals controlling glycogen metabolism.
Right, like epinephrine and glucagon kicking things into gear when you need energy fast.
Exactly. But you mentioned another layer of regulation too, something called allosteric control.
Yeah, that's another really important piece of the puzzle.
So what exactly is allosteric control?
Well, it's kind of like a fine-tuning mechanism for enzymes.
Okay, I'm listening.
So you know how enzymes have those specific active sites where they bind to their substrates?
Yeah, that's where the catalytic magic happens, right?
Exactly. Well, allosteric sites are different. They're like these other little pockets on the enzyme.
Okay, and what happens at these allosteric sites?
Well, certain molecules can bind to these allosteric sites. And when they do, it can actually change the shape of the enzyme.
Really? So it's like a little molecular dance.
It is. And that shape change can either enhance or inhibit the enzyme's activity.
Oh, so it's like a dimmer switch for the enzyme.
Exactly. It's all about fine-tuning that activity based on the cell's needs.
Okay, so how does this allosteric control actually work in glycogen metabolism?
Let's take glycogen phosphorylase as an example.
Our glycogen breakdown enzyme.
Remember how it exists in two forms, the less active B form and the more active A form?
Yeah, and phosphorylase kinase is the one that flips that switch, converting the B form to the A form.
Right, but here's where allosteric control comes in muscle tissue when you're exercising and burning through energy.
Okay.
A molecule called AMP starts to build up.
AMP, what's that?
It's a signal that the cell is running low on ATP, its primary energy source.
So it's like a little red flag saying we need energy stat.
Exactly. And here's the cool thing. AMP can actually bind to glycogen phosphorylase B directly.
Oh, so it bypasses that whole phosphorylase kinase cascade.
It does. And when AMP binds, it activates glycogen phosphorylase B.
So it's like a shortcut to get a quick burst of glucose when it's really needed.
Exactly. It's all about that rapid response to keep those muscles fueled.
But what happens when you're resting and your energy levels are high?
Well, in that case, you don't want glycogen phosphorylase going rogue and breaking down glycogen unnecessarily.
Right. Got to conserve those energy stores.
Exactly. And that's where ATP comes in. When ATP levels are high, it acts as an allosteric inhibitor.
Oh, so it binds to glycogen phosphorylase and keeps it in check?
Exactly. It's like a feedback mechanism, making sure glycogen breakdown only happens when it's really needed.
Okay, so we've got AMP promoting breakdown when energy is low and ATP putting on the brakes when energy is sufficient.
Right. It's a delicate balance maintained by these allosteric regulators.
This is fascinating. So we've got these allosteric controls happening within the cell, and we've got those hormonal signals coming from outside the cell.
Right. It's like two different layers of regulation working together.
But do they ever clash? Do hormones ever override these allosteric controls?
They can, especially when it comes to maintaining blood glucose levels, for example, if your blood sugar drops too low.
Okay.
Glucagon steps in and triggers that cascade we talked about earlier, ultimately activating phosphorylase kinase in the liver.
Right.
But even if a liver cell thinks it has enough ATP, glucagon can force it to break down glycogen and release glucose into the bloodstream.
So it's like the needs of the many outweigh the needs of the few.
Exactly. It's all about prioritizing the whole body's energy balance.
Wow, this interplay between allosteric control and hormonal signals is incredible. It's like our bodies have multiple backup systems, always working to ensure a steady supply of energy.
It's a truly remarkable system, isn't it? And we've only just scratched the surface of its complexity.
I know, it's amazing how much we've learned about glycogen metabolism just in this deep dive.
From those basic building blocks of glycogen to the intricate enzyme reactions, the hormonal control, and now this fascinating world of allosteric regulation.
It's clear that glycogen metabolism is far more than just storing and releasing glucose. It's this carefully orchestrated dance that keeps our whole body running smoothly.
Well said. And with that, we've reached the end of our glycogen deep dive. I hope you've gained a newfound appreciation for this essential metabolic pathway.
Absolutely. This has been a fascinating journey. Thanks for guiding us through it.
It's been my pleasure. Always happy to talk about glycogen.
And to all our listeners out there, thanks for joining us on this deep dive into the world of glycogen metabolism. We'll see you next time.
Bye.
Bye.